Okay, guys. Hope you did well with your information from yesterday. We're going to continue with chapter 11 today. This is going to be a little bit longer, uh, but this is, I guess, what you would call the hard part. But hopefully, after the information that we covered with muscle cells, hopefully action potentials and the generation and conduction of an action potential won't be so overwhelming for you because we've already talked about it once with muscle cells. So it's going to pretty much be the same thing with nerve cells. One big difference again is that nerve cells are not going to start out. Their resting membrane potential is not going to be as negative as with skeletal muscle cells. But other than that, it's very similar to skeletal muscle. So if you struggled with it with skeletal muscle, then hopefully you'll going over it again. Hopefully you'll get a better understanding of it in chapter 11. If you already kind of pretty much have uh, a good understanding of it, then this part will actually be pretty easy for you. Um, so again, a lot of this is going to be stuff that you're going to be like, I've heard this before. Well, you did when we talked about how a nerve cell stimulate muscle cells. When we talked about action potentials in the muscle cell and how it's generated and then how it's what we call propagated. Then you've heard this before in chapter 9. So I hope a lot of this is stuff that you already know and sounds really familiar to you. We may be going over it in just a little bit more detail in this chapter, but it's pretty much the same thing that we said before. So again, remember that an action potential is an electrical signal that is generated in your nerve cells, in your nervous system. It basically is just an electrical signal that transfers information from one part of your body to another. This is how our brain transfers information to our muscles to tell us to move, to walk, to pick up our pencil, whatever the case may be. But it's also the information that travels from our body to our brain to allow us to perceive our environment. To allow us to know if something is hot or cold or is hurting. And then again, of course, how our brain lets us respond to that by stimulating our skeletal muscle, also stimulating smooth muscle and cardiac muscle as well as glands, as we talked about yesterday. So all of the information that's carried from your body to your brain about your internal and external environment, and all of the information that's carried from your brain and spinal cord to your body about how you're going to respond to that environment, all of that information going into your CNS and coming out of your CNS is carried by these action potentials, which are just basically electricity. They're electrical signals. Um, and when we talk about these electrical signals, we have to talk about potential, specifically membrane potential. Remember, we have a resting membrane potential that we'll talk about, and this is when this cell is at rest, when it is unstimulated. But basically, membrane potential is just a measure of the charge difference across the cell membrane. Um, and there's two things that cause this charge difference and these electrical properties of the cell membrane. And these two things are the concentration of ions across the cell membrane. Remember, we have different concentrations of ions on the outside versus inside the cell. We'll look at that more in just a minute, just to refresh your memory. And then also we'll look at the permeability of the plasma membrane. We'll look at how different ion channels allow movement of these ions across the membrane. So again, when we look at that first bullet there, the concentration difference of different ions across the plasma membrane, remember that there is a charge difference on each side of the plasma membrane due to the concentration of different ions. Remember, on the outside of the cell, we have more sodium ions, and on the inside of the cell, we have more potassium ions. Now we also see that we have more chloride ions on the outside of the cell than inside, and we have more negative proteins on the inside than we do on the outside. Again, we have positives and negatives on both sides. It's just that overall, there's more negatives inside the cell and more positives outside the cell. And so this is what causes us to have this membrane potential, this difference in charges, is because overall there's more negatives, more negative proteins and negatively charged substances inside, and there's more positively charged substances on the outside. So when we have more positive on the outside and more negative on the inside, we see that there's more sodium outside and more potassium inside. Even though there's all of these things except for the negative proteins we have on both sides of the cell membrane. It's just that there's an overall difference between the inside and outside. Okay, so that's important to remember when we start looking at these ion channels and the direction that ions are going to move when those channels open. The other thing that we want to look at are permeability characteristics of the cell membrane. The reason that there are differences in the concentrations of different ions inside and outside the cell is due to the different channels that we have inside the cell membrane, in the cell membrane. One of the important ones is the sodium-potassium pump. The sodium-potassium pump, and I have pump there like twice, I don't know why, but anyway, the sodium-potassium pump actually moves 2 potassiums inside for every 3 sodiums it moves outside. So again, if it's moving potassiums into the cell, okay, we already have more potassiums in the cell and we're putting more in there. So it's going against the concentration gradient. It's moving 3 sodiums out. Again, there's already more sodiums outside. So to move more outside, again, it's going against sodium's concentration gradient, and so that process requires energy. So we call this active transport because the sodium-potassium pump has to use energy to pump more sodium outside and to pump more potassium inside. But again, it is the sodium-potassium pump that creates this, that helps to create and maintain this charge difference, to maintain this concentration difference. Because it pumps 3 sodiums out and 2 potassiums in, we end up with more sodiums outside, more potassiums inside, and overall that is one of the reasons why we have this membrane potential, this more positive on the outside with more sodiums outside and more negative on the inside with more potassiums inside. Also, remember there are more proteins inside the cell because these proteins are made inside the cell. They're too large to move out of the cell, and so they remain in the cell. They're large and they're negatively charged, so they contribute to the negative charges inside the cell. Also, chloride ions move out of the cell because they are repelled by those negative proteins. Those large negative proteins inside the cell repel the negatively charged chloride ions, so they move outside the cell with the sodium. Again, we have more sodium and chloride ions outside the cell. And in order to reestablish resting membrane potential after an action potential, and also in order to get depolarization, which creates an action potential, we have to have these ions moving across the plasma membrane, and these ions are going to move across the plasma membrane using two different types of channels, which we talked about back in chapter 3. They're going to use the gated ion channels and the non-gated ion channels. Non-gated ion channels are also called leak ion channels. And those leak ion channels are basically, they allow things to leak out of or into the cell. It's not a large movement of ions into and out of the cell, but it's a little bit of movement of ions into and out of the cell. These leak ion channels are always open, so they're kind of like having little holes in the cell membrane that little things can drip out of. They can just kind of leak into or out of the cell. And they are always open, so even when the cell is at rest, when it's unstimulated, these are still open, allowing ions to move into and out of the cell. Specifically, there are leak ion channels for potassium and chloride, as well as sodium. However, there's a lot more for potassium and chloride than there are for sodium. So when the cell is at rest, you have more potassium and chloride leaking out than you do sodium ions leaking in. Okay, so again, we're going to end up with more chloride and sodium outside because we have more chloride leaking out than we do sodium leaking in. Also, we are going to have some potassium ions leaking out pretty good at rest. But again, remember, even though you have sodium leaking into the cell, you have chloride and potassium. You have chloride leaking out and you have potassium leaking out. Remember, you still have that sodium-potassium pump that is also functioning, even at rest, pumping sodium out and potassium in. So even though you do have some sodium leaking out of the cell and potassium leaking into the cell, uh, or I'm sorry, you have sodium leaking into the cell and potassium leaking out of the cell through these ion channels. Because you have the sodium potassium pumping, it is pumping the sodium that's leaking in back out, and it's pumping the potassium that's leaking out back in. So overall, you still have more potassium inside and more sodium outside the cell. It's the gated ion channels that are going to be most important in generating an action potential, as well as reestablishing, I'm not going to say reestablishing resting membrane potential, because it's the sodium potassium pump that does that. Mainly, these gated ion channels are going to be the most important things at generating an action potential. They are closed until they are opened by a stimulus, and there are different stimuli that can open them. So we have ligand gated, we have voltage gated, and then we have other gated. So you guys got ligand gated are going to be those ion channels, like with our muscle cell, that begin the action potential. They are the ones that, when the neurotransmitter acetylcholine attaches, they open and allow sodium in. If enough sodium gets in to reach the negative 55, which is threshold, okay, then that stimulates our voltage gated sodium ion channels to open. So again, ligand gated begin letting the sodium ions in. They are the ones when, for example, again, in the muscle cell, but also in nerve cells, when a neurotransmitter attaches to a ligand gated channel, it begins letting sodium in. If it lets enough sodium in to get up to threshold, then that opens voltage gated. And when those voltage gated ion channels open, that lets huge amounts of sodium in, and that is what gives us the actual action potential. Okay, so the ligand gated kind of start everything. If a neurotransmitter binds and some ligand gated sodium ion channels open, it lets some sodium in, but if that is not enough for us to reach negative 55, then nothing happens. Sorry about that. That is some stupid phone call. In any case, I hate getting interrupted. But anyway, so ligand gated channels are those channels that neurotransmitters like acetylcholine can bind to, but also hormones can bind to those. And we'll look at those in a minute. And then if you get enough sodium into the cell through these ligand gated channels to get it up to negative 55, which is threshold, then that's going to open up the voltage gated channels. The voltage gated channels are then going to let large amounts of sodium in, which will actually give us that action potential that takes us all the way up to the positive 35 or positive 40. And then the other gated ion channels, you guys, they're going to do the same thing as these ligand gated ion channels. Things like mechanically gated channels. So channels that are open due to pressure, those are mechanically gated. We also have light sensitive. We have vibration sensitive. Again, those are going to be your mechanically gated. But like receptors in your eye are going to have these other gated ion channels. They're going to be stimulated by the presence of light that then is going to stimulate those voltage gated. So the voltage gated are the ones that are going to open once it reaches threshold. The ligand gated and the other gated, like our mechanically gated, our photosensitive gates, they are going to open to allow enough sodium to get in to reach threshold. Once it's reached threshold, it's these voltage gated that are going to open and give you the actual action potential. Okay, so just looking at these different ion channels a little bit more closely, again, the ligand gated are they have a receptor site that is on the outside of the cell, so it's on the extracellular side of the cell membrane, the outside of the cell membrane, so that it can receive these signals from the environment outside the cell. So it can receive a neurotransmitter or hormone from outside the cell. So when the neurotransmitter or hormone binds to the receptor site on this ligand gated ion channel, then it will open. And when it opens, it will allow sodium ions into the cell, hopefully enough to reach negative 55, to go from negative 90 or in a muscle cell, or negative 70 in a nerve cell, all the way up to negative 55, which is threshold. So again, an example here, the sodium ion channel that binds acetylcholine is a ligand-gated ion channel. So when acetylcholine binds to the receptor site on that ligand-gated ion channel, that channel will open and let sodium in. And hopefully enough sodium will get in to bring it up to threshold. There are not only ligand-gated ion channels for sodium, but there are also ligand-gated ion channels for potassium, calcium, and chlorine. So in addition to having leak ion channels for sodium, potassium, and chloride, we also have ligand-gated ion channels for those. So again, lots of ions moving across the cell, both at rest and during stimulation. The calcium ion channels, we'll see a little bit later, will be very important in stimulating cardiac muscle contraction. So again, this is showing you a closed ligand-gated ion channel. Here's our receptor site. That is a receptor protein, usually a glycoprotein. When the messenger, again here, this is going to be our neurotransmitter like acetylcholine or hormone. When it binds to that receptor site, it opens that channel and starts allowing and we're going to say here, let's call the little blue circles sodium ions. So when the acetylcholine or the hormone binds to the receptor site on this ligand-gated ion channel, this is the ligand here, the messenger is the ligand. So the acetylcholine or the hormone, the neurotransmitter of the hormone, when it binds, it opens the channel. Notice here the channel is closed, here it's opened. It causes a change in the shape of the channel so that it's open now and it allows those sodium ions to come into the cell. And so again, if we get enough sodiums in here to bring the inside of this cell from -70 or -90 to -55, then that's going to stimulate the opening of the voltage-gated ion channels, and that's what we're going to look at now. The voltage-gated ion channels are going to respond to small voltage changes across the cell membrane. So again, remember that on the inside of the cell is negative, the outside is positive. But if we open up those ligand-gated channels and get enough sodium in to bring our charge up to -55, then this is going to stimulate the opening of these voltage-gated ion channels. When the voltage-gated ion channels open, then that's going to allow the sodium into the cell. Also, we have voltage-gated potassium ion channels. So if we get up to -55, not only is the sodium going to start coming in, but potassium will start going out. Again, in a cardiac muscle and in smooth muscle, calcium ions are also important. We looked at in chapter 9 how that it's not just the movement of sodium into the cell that stimulates the contraction of the smooth muscle and cardiac muscle, but also the movement of calcium ions into the cell. So, and remember we talked about how the calcium ions bind to calmodulin once they get into the smooth muscle cell, and that begins the muscle contraction. So it's going to be calcium ions, in addition to sodium ions, are going to move into the cell when you reach that threshold, -55, in the cell, and trigger the opening of more ion channels to cause that depolarization, the complete depolarization of the cell. So remember, that is a way that smooth and cardiac muscle were similar, was that they both, not only sodium, but calcium was also important in causing the contraction. Both sodium and calcium move into the cell to cause contraction in your cardiac and smooth muscle. So here we see a voltage-gated ion channel. Here is resting membrane potential. We have negative inside, we have positive outside. When we get enough sodium into the cell to change this inside to more positive, now again, it's not totally positive, but it's more positive, okay? It's reached -55. This causes a change in the shape of this sodium ion channel and allows more sodium ions in. And this is when we're going to get complete depolarization. We're going to go to positive 35 or 40 inside the cell, and the outside will be more negative. Okay, the other gated ion channels, I'm only talking about a couple here. We have touch receptors, which use mechanically gated ion channels. So pressure can cause a change in the shape of the ion channel, allowing it to open and allowing sodium ions in. So like, touch. And then temperature, we also have temperature receptors. They change shape and open in response to a change in temperature. This is going to be different receptors in your skin are going to open as a result of changes in temperature and allow the these ion channels are open due to a change in temperature, they're going to allow enough sodium in to reach the -55, which will then open up the voltage-gated channels again, allowing more sodium which will actually cause the complete depolarization. Okay, so this is just kind of showing you all of them. Here's your leak ion channels. Again, always open, allowing potassium ions out of the cell, because remember, they're diffusing out because there's more potassium inside, less outside, so the potassium diffuses out. And then with sodium, again, there's more sodium outside, less inside during when the cell is resting, and so sodium diffuses into the cell through these leak ions. Again, they're always open. Then we have our ligand gated. This is like the where the acetylcholine, which is a neurotransmitter or a hormone, can bind. Here it's closed, here it's open, allowing that sodium into the cell. Again, it is going to be this or this that is going to allow enough sodium into the cell to open these, the voltage gated. So again, the ligand gated, the neurotransmitter or hormone binds, opens it up and allows sodium in, hopefully to reach negative 55. And then with your mechanically gated, this is going to be touch receptors. This is going to be your sound receptors in your ear. Again, vibrations are mechanical. So vibrations, touch, pressure here causes a change in the shape of it. So when we push down here, this kind of pulls this open and is going to allow, let's say here, sodium ions into the cell. Hopefully we'll reach negative 55, and then that will trigger the opening of these voltage gated. So these two, and in addition, there are others, as I said, the light receptors in your eye, for example, taste receptors in your taste buds on your tongue, odor receptors in the olfactory epithelium in your nose. So there's other types of receptors, but these are two very common ones: ligand gated, where a neurotransmitter or hormone binds, and pressure or vibration, triggering the opening, and either one allowing enough sodium in to reach negative 55. Once you reach that threshold, then that triggers the opening of the voltage gated. The voltage gated open. Again, there's lots of these voltage gated, allowing even more sodium in, which is going to get us our complete depolarization and our action potential. So again, looking at how do we establish resting membrane potential, again, we've already said there is a charge difference across the cell membrane, because there's more potassium and more large negatively charged proteins on the inside. There is more sodium and negative chlorine, or chloride, I should say, on the outside, so that overall the inside has a more negative charge and the outside has a more positive charge. When the cell is resting, when we have this resting membrane potential, inside negative, outside positive, we say the cell is polarized. It's polarized. It's sitting there waiting to be stimulated. We call this difference in charges on the inside and outside of the cell, we call it a potential difference, because there is an unequal distribution of charges. Again, overall, there's more negative inside, more positive outside. Again, in muscle cells, we said that resting membrane potential is negative 90, but in nerve cells, it is negative 70, and that should be millivolts. So again, it all just depends upon what type of cell you're talking about. And then, again, it is due to the fact that there are a difference in ion concentrations on the inside and outside the cell, and it is also due to the fact that there's a difference in permeability of the cell. The cell is more permeable to the potassium and chlorine than it is to the sodium. There's more leak ion channels for potassium and chlorine, so chlorine is leaking into the cell. Potassium is leaking out. Again, there is some permeability to sodium at rest. Sodium is leaking into the cell some, but again, not as much, because there's more leak ion channels for potassium than there are for sodium. And that's what I just said. The membrane is more permeable to potassium because there's so many leak ion channels. So potassium is able to leak out more than sodium is able to leak in. Again, so how do we still have more potassium inside than outside the cell? That's due to the sodium-potassium pump. The sodium-potassium pump is pumping sodium out and potassium in. So that keeps, even though potassium is leaking out, it keeps more potassium in than out. So overall, we have more positives outside the cell, and we have more negatives inside the cell. And again, it is the sodium-potassium pump that is maintaining that the concentrations of more potassium inside and more sodium outside, because it's moving 3 sodiums out and 2 potassiums in. This is just showing you resting membrane potential. Again, these are our leak ion channels here. We can see that there's more sodium, the pink balls on the outside, and more purple balls, which are potassium on the inside. Again, the purples are the ones that are letting the potassiums out. The pink are the ones that are letting sodium in. Notice there's not very many sodium, so there's not a lot of sodium leaking in. Notice there's a lot more potassiums, the purple. So there's a lot more potassium going out. So again, so then why don't we end up with the same amount on both sides? Well, it's this sodium-potassium pump. It is actively using ATP, pumping sodiums out and potassiums in, so that even though we have potassium leaking out at a pretty high rate, we have it being pumped back in by this sodium-potassium pump. And even though we do have some sodium leaking into the cell, we have it being pumped back out by the sodium-potassium pump. Okay, so depolarization, what happens? We've talked about this before. It is due to the diffusion of these ions, guys. Depolarization is always excitatory. It occurs when the inside of the cell membrane moves closer to zero. It becomes more positive. So, like, for example, when those ligand-gated sodium ion channels open, sodium ions diffuse in because there's more sodium outside than inside. Sodium ions diffuse in. And that brings our charge, those positive ions coming in, makes our charge inside the cell more positive. It goes up from -70 to -55, hopefully. Okay, and that's going to open those voltage-gated ion channels. Again, there are more than just sodium ion channels, ligand-gated sodium ion channels that can cause depolarization. Again, sodium is the most common way that depolarization happens. Again, when those sodium ion channels open, sodium diffuses down its concentration gradient into the cell. But there are, and that causes the inside of the cell to become more positive. But there are other ligand-gated ion channels that can cause the cell to become depolarized. Calcium ions. If we open calcium ion channels and allow calcium to diffuse into the cell, then that will also cause the cell to become more positive inside. Take us from a more negative charge to a less negative charge. And cause depolarization. So again, and this is part of what happens in cardiac and smooth muscle. Not only do you have sodium coming in, but you also have calcium coming in, and that brings our charge closer to zero, more positive inside the cell, and causing depolarization. Also, calcium ions, when they are used for depolarization, they actually can stabilize those voltage-gated sodium ion channels. So if we have enough calcium, then our sodium ion channels will remain closed. So they kind of help to stabilize those channels. And remember, we can also have calcium ions that regulate the neurotransmitter being released into the synaptic cleft. If you'll remember, we talked about how calcium ion channels open in the nerve cell. And when that calcium comes in, it stimulates the release. It stimulates the fusion of those vesicles containing acetylcholine with the cell membrane and allows the acetylcholine to be released so that it can bind to the ligand-gated ion channels on the muscle cell membrane, on the motor end plate. So again, lots of roles of calcium, but mainly here we're concerned with the role of it causing depolarization in the cardiac and smooth muscle. And then potassium can also cause depolarization. Again, normally potassium diffuses out of the cell, but if you have changes in the potassium levels in your body so that the extracellular concentration of potassium were to become more than inside the cell, then potassium, rather than going out when depolarization happens, it's going to come into the cell and cause depolarization. And this can cause problems with actually hyperpolarization, and it can cause muscle weakness and things like that, because instead of getting depolarization, which stimulates the muscle cell to contract, you're going to get hyperpolarization, which actually is the opposite of stimulating it. So you're going to end up, if you have too much potassium, you're going to end up with a lot of muscle weakness, not only for your skeletal muscles, but also with your heart. It's going to affect the beating of your heart. Because if you have too much potassium outside the cell, then it actually, when those voltage-gated potassium ion channels are open, potassium is going to come into the cell along with sodium because there's more outside than inside. And so instead of getting depolarization, because depolarization happens when sodium comes in, hyperpolarization happens when potassium comes in. You're going to end up with hyperpolarization, which means no contraction. So this is going to cause muscle weakness. It's also going to cause problems with your heartbeat. And when we talk about hyperpolarization, here's what we're talking about. It's always inhibitory. It is when the inside of the cell becomes more negative, which means it's going to move further away from zero. So, like, for example, if we had enough potassium ions coming in that we went from negative 70 to negative 90, we're going to be less likely to get an action potential. We're not going to be able to stimulate that muscle to contract like we should. And there's two ways that we can hyperpolarize: if potassium is exiting or chlorine is entering the cell. So the most common way that cells become hyperpolarized is when potassium ions go out. They exit the cell. If your gated potassium ion channels open, again, we're looking here at like our voltage gated, but could be our ligand gated as well, then potassium, again, will diffuse out of the cell, and the inside of the cell will become more negative. And when this happens, then you can actually, as I said earlier, it can result in muscular weakness, problems with your heart beating, problems with your reflexes being sluggish, again, because we are letting too much potassium out of the cell, causing the inside of the cell to become more negative than it normally would be. Another way that we can hyperpolarize is if chloride ions enter the cell. Again, anytime you are taking positives out, which is what we're doing with the potassium, or if you're letting negatives in, you're going to cause the inside of the cell to become more negative instead of less negative. And so you're going to, again, cause hyperpolarization, which means further away from zero, which means no action potential, because action potentials happen when we get to negative 55. If we're driving the charge inside the cell down, then we are not getting our more positive charge inside the cell, and we're not going to get that action potential. So again, depolarization is when we go more positive, closer to zero. Hyperpolarization is when we go more negative inside the cell, which is further from zero. So again, taking out potassium or putting in chloride, either way, that's going to bring the inside of the cell more negative, and that is not going to get us an action potential. Okay, what is a graded potential? Okay, a graded potential is what happens when you open up those ligand-gated ion channels, when you open up those mechanically gated ion channels. When you get just enough stimulation that you open up those ligand-gated or mechanically gated ion channels, it lets in a little bit of sodium and or calcium, if we're letting calcium in, like in smooth and cardiac muscle, that we get a relatively small change in membrane potential in just one localized area of the cell. Again, many things can cause this. Again, as I said, chemicals like acetylcholine, any neurotransmitter or hormone binding to a ligand-gated ion channel, a small change in the voltage triggering the opening or closing of voltage-gated ion channels, pressure opening some mechanically gated ion channels, temperature opening some ion channels in your thermoreceptors, or you can just have spontaneous changes in the permeability. Any of these things are going to cause a small change in membrane potential. Again, these small changes, they can be real small, they can be a little bit bigger, they can vary in size depending upon the stimulus that caused the ion channels to open. And they can be hyperpolarizing or depolarizing. What's important about these is that all these little changes can add together to give us a big change, to give us enough of a change in the charge to get us to threshold, which is then going to cause an action potential. So again, if we have enough of a stimulus, if we have a very small stimulus, then it's going to give us a very small change. And that may not end up getting us to threshold, which is going to open those voltage-gated ion channels and let us have an action potential. But if we had several very small changes successively, then those can summate. They can add together, and that will give us a big enough change to get us to negative 55, which is threshold, and give us an action potential. So these little bitty changes, due to the stimulation of some of these different, due to the stimulation which causes opening of some of these different channels, if we can get a little bitty change, enough, not enough to get us to threshold, but if we get lots of them, it can still be enough to get us up to threshold. And then again, we can get a graded potential that is a stimulus that is strong enough to open enough of these channels to let in enough sodium to get us to threshold. So if the stimulus is strong enough, then the graded potential will be enough to reach threshold. But even if it's not strong enough to let in enough sodium ions to get us to negative 55, if we have several in succession, one right after the other, then we can still summate and get a large enough change to get us up to negative 55 to get the action potential. That's what we're saying here. So again, if you have enough of these, even if these graded potentials are very small, if you have enough of them, they can still get your charge up to negative 55 and get you an action potential. These graded potentials, however, don't last very long. They rapidly decrease as they move across the plasma membrane from one area to the other, and so they can't transfer information over long distances. So think about if you have an instructor at the front of an auditorium, and let's say you're sitting in the very back, okay? And that maybe that instructor doesn't have speakers and a microphone. They're just standing up there talking. So by the time as the sound moves across the room, it gets weaker and weaker and weaker. So that by the time it gets back to you in the very back of the auditorium, it's going to be hard for you to understand. So that's what we're comparing these graded potentials to. They're small anyway, and as they move across the cell membrane, as they move across the cell membrane from one area to the other, they just get weaker and weaker and weaker. So therefore, they don't last very long. They can't go very far, and they don't last very long. The ones that last long and go far are the action potential. So these little bitty small changes can add up to give us the big long one. But they themselves can't do a whole lot because they're just not strong enough to do much. Sorry, I'm having to change position here. Okay, so again, this is showing us two different ways that our graded potential can cause an action potential. Okay, we can have stimuli that give us lots of little graded potentials. Notice how this first stimulus, it's very small, the next one, but when we add another one, it gets a little bit bigger. We add another one, so we're summating them, we're adding them. It's kind of like wave summation with our muscle. If we get enough stimuli, we can get up to that complete tetanus. Well, that's kind of what we're doing here. If you get enough stimuli, you can actually get up to threshold and get an action potential if you get enough stimuli. But you have to have, if they're not, like here, we have a stimulus that is strong enough that gets us to our threshold, and we're going to get an action potential. But here, if we have several small ones, but we add them together because they're happening very quickly. And so we end up with a bigger change in charge. Again, we're having change in charge. We can end up with enough change in charge to get us up to threshold, which will give us an action potential. So again, you can have many small stimuli that can summate, they can add together to get you up to threshold and get an action potential. Or if you have a stimulus that's large enough, then it can give you a graded potential that's high enough to get you up to threshold and get an action potential. Again, action potentials are the big ones. They are very long and they can go very far. Graded potentials are very small. They don't go very far. The only way that you're going to be able to send message far is if send the message far is if you have enough graded potentials to get you up to threshold so you can get that action potential. So the action potential is the means by which neurons communicate with their effector. They result from summation of graded potentials. So again, if we get enough graded potentials in succession to get us up to threshold, then we're going to get this action potential. Again, remember that action potentials are going to be generated in the trigger zone. Remember that is going to be the axon hillock and the initial segment at the beginning of the axon of the neuron. So the graded potentials are going to be generated there. They're going to summate in the trigger zone at the axon hillock. And the reason it happens there is because there's a lot more voltage-gated ion channels there. Again, we've got to get those voltage-gated ion channels open to get enough sodium in to get us up to threshold. Again, the action potential, whereas graded potentials are small changes in the membrane potential, which is small changes in charge, the action potential is a large change. So if we have enough small changes that add up to get us up to threshold, then we're going to get this action potential, which is the large change that can propagate over long distances and transfer information all over the body from one part to another. So again, here is our neuron. Here's our axon. Here is our axon hillock and our initial segment, and this is our trigger zone. It has lots and lots of those voltage-gated sodium ion channels, and so when we get enough graded potentials that open up enough sodium ion channels that get enough sodium in to get us up to threshold, then we're going to open up those voltage-gated channels and that are here in this trigger zone, and we're going to get that large change in charge, that large potential, which is the action potential, and it's going to propagate down the axon. And that's what this is showing you here. Okay, an action potential consists of two phases, a depolarization phase and a repolarization phase. Depolarization phase happens, again, when we get up to threshold. That opens up those voltage-gated sodium ion channels. Sodium ions are going to flood into the cell. They're going to diffuse in because there's more outside and less inside. They're going to move down their concentration gradient into the cell, causing the inside of the cell to become more positive, and this is what depolarization is. This in turn causes even more sodium ion channels to open, which is going to let even more sodium in. So again, this is an example of positive feedback. When you get the depolarization, you get those voltage-gated sodium ion channels open, they let sodium in. This triggers even more voltage-gated sodium ion channels to open, which lets even more sodium in, and so it's just a positive feedback loop. And this is what causes in muscles, for example, contraction. And then we have repolarization, and this is where we return our resting membrane potential. Potassium is going to go out voltage-gated channels, and this is going to allow, we're going to remember the sodium-potassium pump is going to pump sodium out. It's going to pump potassium in, but we're also going to have some potassium leaving through those voltage-gated potassium ion channels, and this is going to end up causing a little bit of hyperpolarization. Remember, hyperpolarization is sorry, that's a message from my sister-in-law. It is inhibitory, not stimulatory. So when we have potassium ions going out, we're going to end up with a slight hyperpolarization because those potassium ion channels, they're open, but it takes them longer to close. And resting membrane potential is restored more quickly than those potassium ion channels close. So the sodium-potassium pump gets the sodium out, the potassium in, but we still have those voltage-gated potassium ion channels open, letting more potassium out than we want to get out. And so that's going to bring our charge down a little bit more negative than the negative 70. And again, that lasts for a very short period of time, but we do have a little bit of hyperpolarization, which is called after potential, there right after the resting membrane potential is restored, because those voltage-gated potassium ion channels close a little bit slowly. It takes them a little bit longer to close, and so they let a little bit more potassium out than we want to. So we actually get a little bit more negative inside the cell than negative 70, which is a little bit of hyperpolarization. Again, just like with muscle, we have an all or none principle for our action potentials in our nerve cells. If the stimulus or the stimuli produce enough graded potentials to get us to threshold, then all that we just talked about is going to happen. Again, those sodium-gated ion channels or those voltage-gated sodium ion channels are going to open, going to let in lots of sodium, and it's going to be propagated, and you're going to get an action potential, and you're going to get the message sent all the way down the neuron. And it will not be interrupted. It will not stop. And that is the all part. If the stimulus is weak enough that it doesn't, if we don't produce enough either a large enough graded potential or enough small graded potentials to get us up to threshold. Then the resting membrane potential will return, and there will be no action potential, and that's the none part. So you either get an action potential or you don't. If the stimulus is large enough or we get enough stimuli to produce enough graded potentials to get us up to threshold, then we get an action potential. We get depolarization and then repolarization. If we don't get a strong enough stimulus to give us a strong enough graded potential or enough small graded potentials to get us up to threshold, then nothing happens. Everything returns to normal, and it's like nothing even happened. Okay, so here we're showing, here we get our graded potential or many graded potentials to get us up to threshold. Once we get up to threshold, those voltage gated ion channels open, sodium rushes into the cell, and we get all the way up to negative 35, or I'm sorry, positive 35 or positive 40. We get that action potential, and then we restore resting membrane potential with that sodium-potassium pump. Again, those potassium ion channels remain open a little bit longer than we need them to, so we get a little bit more potassium going out than we want, so we get a little bit of after potential, but then once those potassium ion channels get closed, then we get back up to our negative 70. Okay, so again, this is just going through all of that process again. Right at the beginning, we are resting. We have more negative inside. We have more positive outside. More sodium outside. More potassium inside. Then, and we see at this point, the sodium ion channels are closed. Most of our potassium ion channels are closed, and so we have the negative inside, positive outside. Then we have depolarization to happen again. A graded potential or enough small graded potentials occur to get us up to threshold here. The sodium ion channels open, potassium ion channels open. Sodium, a lot more sodium is going to move in, the potassium is going to move out, and so we're going to get more positive inside the cell. We're going to get up here to positive 35 or positive 40 inside the cell and get that action potential that will be propagated down the neuron. Then repolarization will happen. Our sodium ion channels will begin to close, and our potassium ion channels will open and let potassium ions out. And basically what we're going to have here is we're going to have the inside of the cell returning more negative, the outside of the cell returning more positive. And that's what we're getting here. We repolarize, but again, it takes a little bit of time for those potassium ion channels to get closed after we restore resting membrane potential. So that brings us down here to this a little bit of hyperpolarization because it takes a little bit of time to close these to stop that potassium from going out. But once they are all closed, then we get back up to our resting negative 70, which is right here. Again, the potassium ion channels are closed, and our sodium-potassium pump is pumping our sodium out, is pumping our potassium in, getting us back to that negative 70 where we need to be. We do have a refractory period that prevents our action potential from going in the wrong direction. Again, we want our action potential to go toward the axon terminals at the end of the neuron. We don't want it going backward toward the cell body and the dendrites. What prevents this? Well, we have an absolute refractory period. Basically, what this means is that once that action potential has been generated in one part of that axon, then there is an absolute refractory period in that part of that axon, meaning that it is completely insensitive to another stimulus. That another stimulus is not going to allow more sodium ions, is not going to open up channels and allow more sodium ions to come in to do another action potential. So another stimulus is not going to cause anything to happen in that part of the neuron for this period of time. And it actually that period of time begins when the action potential and lasts until the end of repolarization. So again, the absolute refractory period begins here and it ends right here. So again, during this period of time, you have no, pretty much that part of the axon is not going to be able to be stimulated again. And so therefore, that is going to keep the stimulation going down toward the axon terminal and keep it from going backward this direction. And then we also have a relative here. So here, you can still have a stimulus that could generate another action potential, but it's going to be a lot harder. It's going to take a really strong stimulus to generate an action potential during this part of the process. So during the hyperpolarization and return to resting potential, you have a little bit of a really strong stimulus here could cause another action potential, but it would have to be a really strong stimulus. But during this part, during this depolarization phase and repolarization phase, you're going to get no stimulus. It can cause another action potential here. And again, that's going to keep the stimulus, or I'm sorry, the action potential moving toward the axon terminal end of the neuron. This is also very similar to what we talked about in muscle cells. A subthreshold stimulus does not cause a graded potential that is strong enough to give us an action potential, or else it doesn't cause enough graded potentials to give us an action potential. So again, nothing happens here. A threshold stimulus is strong enough to give us an action potential, and we get one action potential. A maximal stimulus is strong enough to produce a maximum frequency of action potentials. So again, we can have different sizes of graded potentials, but once you get an action potential, all action potentials are the same strength. Okay, they all go up to positive 35, positive 40, and that's as far as they go. So they're all the same strength. So a maximal stimulus is not going to give us a stronger action potential. What it's going to do is it's going to give us more action potentials, a maximum frequency of action potentials, so very fast action potentials. A submaximal stimulus is going to be a stimulus that gives us any frequency of action potentials between threshold and maximal. So more than a single, but less than maximum. And then supramaximal stimulus is a stimulus that's stronger than maximal. However, even if that stimulus is stronger than maximal, it's not going to give us any greater frequency because we can only have a maximum frequency. So even if we have a stronger than maximal stimulus, we're still just going to get that maximum frequency of action potentials. And I think this diagram shows it to you pretty well. Again, if the orange line is an action potential, subthreshold, we don't get one. Threshold, we get one. Submaximal, we get more than one, but less than maximal. So here's maximal. This is the greatest frequency we can get. Supramaximal, okay, it is a stronger stimulus. Okay, it's not a stronger action potential. Notice that all these action potentials are the same. They're going up to positive 35. So it's not a stronger action potential. This is just a maximum frequency. And notice supra, even though it's a stronger stimulus, notice it's a little bit higher. This is the stimulus. The action potential is still the same. And notice the frequency is still the same because we cannot get more than maximal frequency here. So maximal stimulus gives us maximal frequency. Supramaximal stimulus is a stronger stimulus, but it doesn't give us any more than maximal frequency. We still have the maximal frequency. And notice again, all of these action potentials are the same. What we're changing is the frequency, how often we have an action potential with these different stimuli. So a stronger stimulus can give us a greater frequency. So therefore, what's the difference between a prick in your finger and getting stabbed? Well, a stronger pain is we have a higher frequency of action potentials. So a little prick with a little needle gives you a certain frequency. Okay, it's giving you a submaximal frequency. But a stab, being stabbed, maybe that is a maximal stimulus giving you the maximal frequency. So stronger pain is a higher frequency. So, this right here might be getting pricked with a needle, and this might be getting stabbed. Okay, so again, the pain is stronger because we don't have stronger action potentials. We have a greater frequency of action potentials. Higher frequency action potentials also produce stronger muscle contractions, cause our glands to secrete more. And also the length of action potentials can also affect the amount of pain that we feel or the strength of the contraction, the length of the action potential. Again, going back, looking again, we've already talked about this before, propagation is like dominoes falling. Okay, you don't have the same action potential that goes all the way down the neuron. You have different action potentials. One action potential stimulates another one right next to it, stimulates another one right next to it, stimulates another one right next to it, all the way down the axon, just like dominoes. So when you cause a domino to fall, it's not one domino all the way down the line. One domino causes the next one to fall, which causes the next one to fall, which causes the next one to fall. That's the way action potentials are. Again, action potentials only go in one direction, from the trigger zone down the axon toward the axon terminal. They cannot go from the trigger zone back up toward the cell body and the dendrites because of that absolute refractory period. Once you have an action potential in that trigger zone, okay, then the action potential has to move down the axon toward the axon terminal. It cannot move back up toward the cell body and the dendrites because you have that absolute refractory period. In that trigger zone, once it's generated one action potential, it cannot generate another one until that repolarization phase is over. And so that one action potential stimulates an action potential next to it in the axon, and then next to that in the axon, and it keeps it from going back the other direction. Now we have two different types of conduction. We have continuous conduction and saltatory conduction. Continuous conduction happens in unmyelinated axons. Basically, each section of the cell membrane of the axon conducts an action potential. So again, just like the dominoes, one here, then the next one falls, then the next one, then the next one, all the way down the axon to the axon terminals. Again, the action potential does not go backward. We get a one-way conduction all the way down the axon. So here we see it. Again, the orange is the action potential. So the action potential here stimulates one here, and then here, and then here. Again, we can't go back here because this is in that absolute refractory period. So it can't go back this direction. It has to go this direction. Again, like dominoes falling all the way down the axon. The other type is saltatory conduction. Saltatory conduction is used by myelinated axons. Remember that myelinated axons, that myelin is not continuous. It has the myelin, the Schwann cells producing the myelin, and then there is a gap here called a node of Ranvier. Now again, these little globs here, these Schwann cells with the myelin, those are called internodes, and the gaps between them where there is no myelin are called nodes of Ranvier. And basically what happens here is the action potentials leap. So rather than going all the way down the axon, which takes longer, the action potentials leap from one node of Ranvier to the next, just like we can see here. It leaped from here to here, and then from here to here, and then it would leap from here to here. Okay, so the local currents, the action potential flows from one node of Ranvier to the next node of Ranvier. So it leaps. It's like it's leaping. So it's not going all the way down the axon. It leaps from one node of Ranvier to the next. And this is more rapid because the action potentials are what we might say leaping. I have that in quotation marks there. And again, it's faster because there are really high concentrations of sodium, of voltage-gated sodium ion channels here in these nodes of Ranvier. So that also allows it to leap. And so, guys, I have heard it compared to, imagine that you're in a class of 30 people and you have a battery. And if you pass the battery from one person to the other, down the one row, up the next row, down the next row, through all 30 people, okay, that is continuous conduction. Okay, so that is this. That is what your unmyelinated axons are using, okay? And that takes a certain amount of time. Now imagine that we have a battery and we're going to throw it from the front of the row to the back of the row, back up to the front, back to the back to the front, and everybody's catching it, and it's leaping from the front to the back, from the back to the front, from the front to the back, from the back to the front. So it's leaping, and everybody's catching it just like they should. That is saltatory conduction, and I can guarantee you, as long as everybody's catching the battery, as you throw that battery from one person to the next, from the front to the back to the front, back and forth, it's going to get to the other side of the room a lot more quickly than it would if we just passed it from one person to the next all the way through the crowd. So that is a good analogy for continuous versus saltatory conduction, and explaining why it's faster in with the myelinated axons and the action potential leaping or jumping from one node of Ranvier to the next. That's my mom. Sorry, guys. Okay. So what are some things that affect the speed of conduction in a neuron, the speed of the conduction of the action potential? Well, first of all, again, as we just saw, it happens faster in myelinated axons as compared to non-myelinated due to that saltatory conduction. Also, the more myelin you have, the faster the action potential will be conducted. Finally, the larger the diameter of the neuron, the more quickly the action potential will be conducted. Again, because large diameter axons have more surface area, they have more voltage gated sodium ion channels, which is going to allow for a faster conduction of that action potential. So, just like we had different types of muscle fibers, and those were not based upon action potential conduction, but were based upon the size as well as the ability to conduct or the ability to do aerobic versus anaerobic respiration and to produce very strong contractions versus very weak contractions. We also have 3 different types of nerve fibers, again, based upon whether they're myelinated or not, based upon their size, and based upon how quickly they conduct action potentials. So type A are our large diameter myelinated axons, nerve fibers. These are going to be conducting action potentials very quickly. And these are the types of nerve fibers that innervate our skeletal muscles. Also, our sensory neurons are these type A large diameter myelinated. So this means they're going to conduct action potentials very quickly. Why is it important to have these skeletal neurons as well as sensory neurons being type A, conducting action potentials very quickly? Guys, these determine our response to the environment. Okay? Our sensory neurons are able to conduct that action potential very quickly to our CNS, and then our CNS is able to send that action potential for a response very quickly to our skeletal muscles to allow us to react. So, for example, we're about to touch a hot pan. Sorry, that's my dog howling out there. I'm sorry, there's so many interruptions. Anyway, so we are about to touch a hot pan, okay? That information that is very hot needs to be conducted to our CNS very quickly, and then our CNS needs to be able to send a response information signal back very quickly to get us to move our hand. So that is why we need the very large diameter myelinated type A fibers controlling that. So our sensory neurons send that information very quickly to our CNS. Our CNS is able to send the information to respond very quickly back to our skeletal muscle to have us to move. This limits the amount of damage that we're going to get. It allows us to respond by moving our hand very quickly. So I hope that makes sense. Type B and type C, these are going to be smaller nerve fibers, less myelinated, conducting action potential slowly. Why? Because they innervate our internal organs. They allow us to maintain homeostasis, and this response doesn't need to be as quick as those that allow us to sense and respond to our external environment. So again, it's okay if it takes a couple seconds longer to open up those blood vessels so that you can cool down when you're too hot, or to close those blood vessels when you're too cold to try to help you warm up, because you're not going to die in a couple of seconds. You're not going to get severely injured by it taking an extra second or two for those blood vessels to open or close. Okay? With your touching that hot pot, you're going to have a lot more damage, serious damage, if it takes an extra second or two for you to take your hand away from that hot pot. So again, the type B are medium diameter. They're kind of in the middle, light myelination. They're going to conduct action potentials more slowly than the type A. The type C are even smaller, they're unmyelinated, and they're going to conduct action potentials even slower. Again, both of these are going to be the axons, the neurons, the nerves that stimulate that our internal organs, our heart, our intestines, our bladder, things like that. Because it's okay for those responses to be a little bit slower, because we're just maintaining homeostasis there, and it doesn't control how badly we're going to be injured or whether we're going to live or die. Okay, I hope that makes sense. Please let me know if you have any questions. I know there was a few places there when I was talking about the opening and closing of the potassium ions and the moving in and out of the potassium ions that was a little bit off. But again, overall, potassium ions are going to be inside the cell, and they are going to move out when during repolarization or after depolarization, or right there during depolarization. They're going to move out, and then that is what causes that hyperpolarization. They actually move out too much, and then resting membrane potential is restored. Please let me know if you have any questions. I hope a lot of this was just review from our unit on muscle contraction. And again, you can email me or send me a message on Google Classroom if you have any questions or have any trouble. And we'll talk to you later. Have a good day. Bye-bye.